We're gonna do organoids.
Okay.
Alright, so we are going to start by having SciShow introduce us to the subject. This is an old video. This is 5 years ago, this video. So I'm going to drop in the updates probably after calling them mini brains for the entire rest of the video. I'm going to try not to be too annoyed by it. But I'm going to just say this one time. They're not mini brains. Scientists do not refer to them as mini brains because that would be grossly misleading. They're organoids. They are brain organoids.
All caps, she even said this.
We're stopping everything. What can we do for you?
Oh, Ian asked what about mini stomachs? I think, well, they are expanding to different organs beyond what she has been saying. The hearts are now, right?
I mean, I think the first one was mini gut. Wasn't that the first organoid ever built was part of an intestine?
I think you're right. Yep.
I'm pretty sure that this is a thing. Okay. I'm going to just point out she's calling this a cerebral organoid because most of the brain organoids that have been created take after the cerebral cortex. There are different parts of the brain, and what scientists are discovering is you can customize your organoid to more closely look like different brain regions. So the one that made the news recently that there was a big paper on was the cerebellum. Okay, so anyway, it's not a mini brain. She's going to keep calling it a mini brain, but it's not a mini brain. I'm going to call some mild shenanigans on this. I feel like she's not doing right by the rat and the mouse models here, but fine. They're not human. They're not the same as human cells. Fine, because they're a mouse cell and a rat cell. They are quite similar, but I am not saying she has a mini brain. I'm suggesting that a mini brain is not a thing. Therefore, I doubt she has one. Okay, so this is my general issue with the term mini brain. Is partly what she just mentioned. They're not forming directed connections the way brains do. They don't have the same networks. They don't have the same complicated structure. They are little globs of neurons and some other cells. I like organoid. I think it's a fair. It makes you think that it's similar to an organ, right? It's just not a fully functional thing. Especially when it comes to brain, I do start to worry. She had mentioned this in the video earlier, the question about ethics, and I do think there might be a point at which we can start arguing about what's the ethical threshold for growing human neurons in a dish. I would suggest we are not at that point. Like these neurons can send electrical impulses if they're prompted, but they don't communicate, and they're not the way a human brain does. They're not structured in the same way, and it's also kind of random. How it is that they grow, because it is sort of this like random collection of neural tissue. So it's just very different. It's very different, and I worry that if we're calling it a mini brain, people are going to start thinking that it is something that it's not. The technology is somewhere that it's not.
So Andrea says it's just brain cells and not complex working whole brains. Mini brain would suggest.
Yeah, and to be clear, it is like an advancement. Like I've never grown brain organoids. I mean, for my purposes, I've never needed to, and so I've just done what a lot of people do, which is grow single layers of neurons or sometimes a couple layers of neurons, and that's good enough for my purposes. And so this is this is something more complicated. It is a 3-dimensional network, but it is very far from anything that would resemble a brain.
And then I liked Hogan's comment, the stem cells were giving structural support. And after time, they went to college and got degrees in liberal arts.
Yeah.
Allegedly.
Okay. Okay, so I think the mini brain term could have been used so regular folks can understand it somewhat. Yeah, I I 100% get that they're trying to make it more accessible. And I do think that they tried to make it clear that they're not actually miniature brains. I'm not sure that I agree with their choices. But yes, I definitely agree that they chose that phrase, I suspect, instead of brain organoid, because to help relay the concept. Does growing them in a dish versus a fluid like a fetus grows and make it harder to get the layers they need? So I think the biggest issue, there's a lot of issues, but as far as size limitation goes, it's actually vascularization. I think that they did talk about this a little bit in the video. So they can grow, they don't necessarily have to, they don't grow like attached to something necessarily. These organoids kind of grow in this like matrix gel. But at a certain point, you have so many cells that if you don't have blood vessels moving through it, those cells in the very center can't get that nice juicy fluid that has all of the energy and nutrients they need. And then those cells in the center will start to die. And then instead of having a nice complex structure, you just have like a sphere of live cells on the outside and dead cells in the center because they can't access all of the nice juicy stuff that they need that's floating around in their brothy soup. And so scientists are working on that. I don't think that's not like the biggest limitation in my mind. Anyway, all right, so this is an older video. What was this? 5 years ago. This video was 5 years ago. So let me tell you about the newest study. Scientists have gotten a lot better at making brain organoids that resemble different parts of the brain. And this most recent study looked at the cerebellum, and that's really interesting for a couple of reasons. And I'll show you this like one cool cell type that they were able to get. But okay, so why does this matter? Well, this is a specific brain region that's been hard to study, so people are excited for that reason. But I think it's also kind of interesting because of the way the technology's been developing. So, if you recall, the way these brain organoids are made, let's see, I think they had, there we go. These are the organoids. Right here. These little guys floating around in a dish. Can I just say, like, look at the size difference between these things? Like, there's not a lot of consistency in what you're going to get. So there are a lot of limitations, but okay. Yeah. And so scientists are getting a lot better. They haven't been able to grow a complex organoid that resembles all of the different parts of the brain, but they're getting good at growing brain organoids that resemble one part versus another. So this recent study quasi-perfected the cerebellum, like I said, which is usually something just thought of for like motor coordination. But there's a lot of evidence. They have some really important cell types that are difficult to study. And there's some evidence that actually autism spectrum disorders have a lot to do with some of the important cells in the in that brain region. Okay, but I wanted to talk about this because these scientists are trying to do something unique and kind of cool. So to get these little stinkers, these brain organoids, you have to start with a human stem cell. And a lot of people freak out when they hear human stem cell because the first thing that your mind goes to is embryonic stem cell. Okay. That's not what these scientists are doing. These scientists are taking skin cells, for example, any adult tissue, but usually we think of skin cells. They can reverse engineer them into a stem cell, and then from your skin cell grow a brain organoid. And so these scientists, this group and a couple of others, what they want to do is develop this technique into personalized medicine. So different diseases present different, like, there can be a genetic disease, right? Let's say Belinda and I both had a genetic disease. My presentation of it could be very different than his. And that might not necessarily be due to the mutation that's giving us the illness. It might have to do with other little quirks in our genome that make us different from one another. And so the idea that these scientists have is we have the technology to take a skin sample, turn it into a stem cell, and from that stem cell we can grow pretty much any organoid, any type. So they're interested in neurological problems, whether they be like illnesses or developmental problems, and we can actually study with the particular genome exactly what's going on with that individual. So if it was something that happened during development, obviously we can't rewind the clock. That's something we'd be able to observe as the organoid is developing, right? And get a glimpse as to what happened during development. And then also, like I said, you can do it in the genetic background of the individual. So if I wanted to look at a mouse model to study a disease, let's say all people with disease X have one certain mutation. So I can recapitulate that mutation in a mouse and study it in a mouse. But like I said, everybody has these little quirks in their DNA that makes us different from one another. And sometimes those can contribute to the disease state and we don't really understand why or how. And so what a lot of these scientists are doing is trying to develop a form of highly personalized, specialized medicine. So understanding if I have a neurological disorder, not just understanding what gene is contributing that, but also looking at differences in my genetic background, like all of the other genes that are playing a role, and seeing if that is going to change how the disease is presenting in me. And they can do that now in a petri dish. And I think that that's really cool.
Dacum just wants clarifications that reverse engineer skin cells into stem cells is being done now.
Oh, yeah. That we've had that technology for a while. Yeah, we've had that for quite some time.
So is this just now, is the fact that it's personalized?
Yeah, so the idea is they're taking these, the hope that these scientists have is, number one, they're figuring out ways to study different brain regions, and that was really one of the big factors in this study that came out. This is sort of why it was big news and exciting to nerds like me. But their overall goal, their overall project goal is to take these different technologies that are already out there and kind of combine them to make personalized medicine. So we've been able to reverse engineer a stem cell for a long time. But the idea here is take that technology that we have and apply it to patients, and then find a way to grow, so people that have a certain kind of epilepsy maybe, or this lab was very interested in autism, for instance, or it could even be people who have different presentations of Alzheimer's disease, right? There's a lot of different neurological conditions you could study. Take a skin sample and grow an organoid that resembles the specific brain region that you're interested in. And then you can study what is happening to their neurons in a relevant part of the brain. Right? That's crazy. I think that's crazy. I thought it was really cool. As far as getting it to actual personalized medicine, I would say the cost. It's always the cost, right? This stuff is expensive. And then I would say, again, it sort of depends on what you're going for, but I think the next really big hurdle is once we have the capability of really fine-tuning all of the different brain regions, like growing an organoid for every little brain region that we could be interested in, finding a way to combine those. Because a lot of times the disease state isn't just happening in one region of the brain. Maybe the problem is how one part of the brain is talking to another part of the brain. And so I think that's going to be one of the biggest final hurdles. And when we start talking about that many cells, people are going to be concerned again about, are all of the cells getting all of the nutrients that they need? Is there a way to irrigate them, as it were? Can we do some kind of fake introduction of blood cells or something so that they're all happy and healthy in their little soupy broth? So there are these cells, the Purkinje cells. And the reason I wanted to bring them up is because this is one of the cell types that these scientists were able to make in the the brain organoids that they were making. And they are really beautiful, and honestly, I think that they're what a lot of us think about when we think about neurons. All of these classical drawings of these neurons with these crazy dendritic branches. So they have think of crazy Kevin hair. They have just insane, they're just massively connected to an incredible number of other cells, and they're they're a very specific kind of neuron, and they do something very important. They haven't been able to be made in a brain organoid before, and in this study they were able to make these cells. They just naturally developed and connected with the other cells in that organoid. And so that was one of the big breakthroughs. And I think they're just pretty. And honestly, I think when we think of neurons, I think these classic drawings are one of the things that we think of. Right, Belen, would you say?
Yeah. And there's, I've seen the images, too, ones that actually look like this. And it's really, really cool.
Yeah, so this is the cell type. It's one of the big classic, I think very pretty traditional looking neurons, and they demonstrated that they were able to make these cells in the organoids that they were working on.
Andrea likes that you commented as crazy Kevin hair.
Yeah. Well, here we are. All right. So, just to round out, we don't have to watch this whole video, but this particular video actually had a few images of making the organoids in a lab.
Are those the ones that can get really long, meters in length? The Purkinjes?
I think those are the ones where you're usually tracing their axons. I mean, if you were to line up their little branching dendrites, maybe, but they don't have the dendrites themselves are more web-like. They're not a long snaking axon. I think those are usually interneurons that we're thinking of that connect out to the peripheral nervous system is usually what I think of when I think of those really long crazy guys.
Rose is wondering, why were they able to make this one work? Is there something specific about it?
Yeah, so they just worked out. It's just kind of heroic science. They just figured out the conditions. It's very specific conditions to grow these guys in. So some of the basic technology was the same. Get your stem cell, get your skin cell, turn it into a stem cell, and go through the process of turning it into an organoid. But then you have to signal to the neurons. I think the idea is they just had to work out the very specific conditions, and you have to do this every single time you want to create a different brain region. And so they were able to, I think, partly get lucky, because a lot of science is getting lucky, and also just power through to actually optimize to create an organoid that looks like this brain region. It's just an incredible amount of elbow grease, really.
So to get another one of these organoids to work, you'd have to. It wouldn't necessarily be translatable from this, right? You have to work out the specific conditions.
Yeah. For each brain region, you have to work out the specific conditions. But there have been a bunch of groups working on different brain regions, so it is getting pretty comprehensive. But this is the brain region where those Purkinje cells are found, and so it was really reassuring to see that not just that those cells were there, but that they looked the way we expect them to sort of traditionally look.
Which is different from previous, right? Like the previous video where it was just like...
Yeah, this is a totally different brain region, and honestly, it's just the OG brain organoids just kind of looked like vague cerebral cortex, just kind of a shoulder shrug, like, yeah, it's kind of brain-like. And so the technology is advancing so that they can really start to pinpoint and fine-tune to different brain regions.
Is it drastically different between the brain regions?
Yeah. I mean, there is some shared elements, certainly, but then different brain regions, I mean, they have different structures, and a lot of times they do have neurons that are kind of more specific to that region. I think that organoids are probably a long way away from answering some of these, like, larger questions, but we're moving in the right direction, and I think that that's what's really extraordinary. And I love that this particular group that was able to develop brain organoids that looked like the cerebellum, they're very interested in studying disease models coming from human beings that have neurological ailments of some sort. That's one of the things that they're actively trying to work towards, and so I think it's a beautiful step towards personalized medicine. I think we're a long way away, both from the technology and also the financial side of things, but I think it was beautiful and pretty cool research that's going on. Your beautiful, amazing brain has more than 1 billion neurons in it, and these little guys floating around here have a couple thousand. And they're not as well connected.
But it's a way to start that model.
Yeah, it's a way to start, but your brains are big and beautiful and very well connected. They know all the right people. All right? These little guys. So I think it's a move in the right direction. I think it is. But I think for something like that, we need the more comprehensive brain organoids. I suspect we need something where the different brain regions can start to be brought together. And right now, scientists are working towards being able to create an organoid that resembles one brain region, and then the next. And hopefully once we can create all of the different regions of the brain, they'll be able to find a way to unify them. And then we're in business, chat. Then we're really in business. Okay, so we can just watch a little bit of this video. I just thought it was cool because they actually bothered to show you the information itself. Will probably be repetitive, but I like that they actually show you what these things look like and how some of this stuff happens in the lab. Specialized brain organoids versus vascularized. So I think they need both, right? So at a certain point, so chat, for those of you who haven't been following along, one of the limitations to these organoids, there are many, but one of them is eventually you have so many cells that the cells in the very center aren't getting all the nutrients and the happy broth that they need, and they will start to die. So the way your brain gets around that is it's vascularized. It has blood vessels bringing all of the good stuff to your brain. And, well, okay, so blood vessels, but also your brain is not just like a solid mass, I guess, is one of the more critical aspects. And we right now don't have any way of these little brain organoids are just a cluster of cells. There's no complicated beautiful folding and channeling inside these structures. So the guys that are in the center are just totally isolated and don't have access to all of the good Subi nutrients that they need. And so if scientists were to start assembling organoids, yes, one, they would need to be able to have all of these different organoids that are specialized to look like your different brain regions, and then also they would need to find a way to make sure that they have the ability to keep all of these cells alive. So whether it be a more complicated three-dimensional structure or some kind of tubing, or who the heck knows how they're going to work that out.